Yeah, it's not worth it when it's the last one. Yeah. Can you throw in the clicker? He's just gonna have me move the slides anyways. So.
Hello, thank you for coming to my presentation. My name is Bradford Norman. I'm the Wetlands Program Coordinator for the Resighini Rancheria in Klamath, California. Next slide. I'm used to doing these presentations with a laser pointer, but it doesn't work on this type of screen, so I might come up and have to use my hands. So a little project background. Resighini Rancheria is nestled along the Klamath River in the Yurok ancestral territory of northwestern California. It has abundant natural resources, including wetland nursery habitats for important fish species, coho, salmon, amphibians, birds, carnivores, etc. And in 2017, Resighini Rancheria received the Wetlands Initiative Program Grant. I think it was $230,000 to start a wetland program for the tribe. And the first 2 years of this presentation will cover that area, and then the next couple of years will be used to develop our program and do some more in-depth studies in the wetlands. So next slide. So here's some context. We're at the mouth of the Klamath River where the red star is. The tan is the Klamath drainage. We're at the Oregon, it crosses the Oregon border. The inset shows kind of the area that here. That's the map. And Yurok ancestral territory extends from approximately north of where that Klamath is and south to Little River, which is in Humboldt County, and then inland along the Klamath River to, just east of Wichita. There's a big rock in the river that's called Big Rock, and that's the boundary between the ancestral territory for the Yuroks and the Karuks. And the watershed area of the Klamath Basin is approximately 15,700 square miles. The Klamath River extends 286 approximate miles from the mouth of the Pacific Ocean all the way up into Oregon and the headwaters for the Klamath Falls. There's the largest dam removal project in the United States and maybe in the world that's going on right now. One of the dams has already been removed, and 3 of them are set for removal next year. This year and next year, they're doing the drawdowns on the dams, and then the dams are approximately from between the Shasta River and where the Keno area up there. The rancheria itself is about 455 acres of tribal trusts and fee lands combined. The tribal enrollment is approximately 150 members, and there's a wide diversity of habitat types, including riparian, the main stem of Klamath, floodplains, gravel, bars, temporary creeks, permanent creeks, grasslands, willow, cottonwood forests, and redwood, mixed hardwood forests. Next slide. So here's a aerial photo of the area of the rancheria. Up the inset is the boundaries. I think the stippled lands are the fee lands, and the non-stippled are the trust lands. North is at the top. Klamath, the township that was pretty much wiped out in the '64 flood, is over here, upper corner, Highway 101 bridge with the golden bears. People stop and take the pictures there all the time. Tribal offices, Wachtel Creek coming down 101, adjacent to 101, comes through the rancheria for a while, and then goes out to the over here at the main stem of Klamath. We have Junior Creek is a very important non-natal coho rearing area. They come in the winter and feed, get double in size, and go out in April and May, and start going back out to the river and out to the estuary as smolts. The river is running west in this direction, and we have a gravel extraction area on Wackel Flat that's permitted by the Army Corps of Engineers. We have a gravel pit in this section, floodplain ponds up here. In the winter, the river will rise often and flow through that Wackel Flat and through those ponds like it did last year. And then seasonally, the river will go down and we'll have the isolated ponds again. Next slide. So what is a wetland? Three defining factors are the hydrology. It has to have the water, has to have hydrophytes or water-loving plants, and those plants could be both obligate wetland plants, so they have to have a wetland to grow, or they could be facultative wetland plants where they can do well in water, but they can also be upland plants. And then hybrid soils that create anaerobic conditions in the soil is a determining factor for wetlands. And we don't do a lot of hybrid soil stuff, but our tribal capacity isn't at that point yet. But next slide. So the EPA has a definition, and I'm not going to read all this, but there's also Army Corps of Engineer definition. There's also Fish and Wildlife definitions using Cowerman et al. In 1979, I believe it is. But they support both aquatic and terrestrial species, and a prolonged presence of water creates conditions favor the growth of specially adapted plants or hydrophytes. Next slide. So the first stage of our wetland program development was the Quality Assurance Program Plan, or the QAPP. It's a very technical document. It's complicated, time-consuming. It took about a year, I think, to get it done. I suggest if your tribe is interested in doing a wetlands project, a wetlands program, and you'll have to do a QAPP to meet the EPA standards. And I suggest you review existing regional tribal QAPPs, get some ideas. Use templates and seek EPA consultations if possible. So you do your QAPP, you draft it, send it to the EPA. They'll do edits and comments, and that could take as long as 3 months to get it back or longer. And then you'll do your revisions. The tribal council will approve the revisions and send it back to the EPA. They'll do some more comments, and then hopefully you'll get the draft approved from the EPA, and then your tribal council will ultimately have to approve the QAPP. Next slide. The second stage we did is we conducted a wetlands inventory and mapping, and it consisted of site names, which I used topographic maps, Google Earth, aerial photos. Then I GPS the locations, or mostly just access sites using handheld GPS receivers. I used UTMs because they make more sense than the longitude, latitude. And then all of California is in, when you use your GPS, you're using zone 10. I used an Excel file, so the inventory consists of site numbers, location descriptions, wetland types, the GPS locations for the access points, including elevations, which is a little bit problematic when you're using those handheld GPS units. But if you have enough satellites in your constellation and you let it cook for a longer period of time, you can usually get the elevations pretty accurately. And then we did digital photographic documentation, and we had for the Fish and Wildlife Service previous survey of 2006 that already had a lot of our sites mapped. Next slide. So here's some examples of the wetland sites on our reservation. Wackel Creek. We have some unnamed trips that come down into Junior Creek, but I gave them names based on tribal members and land assignment names. We have some springs that we can't see them on the map, but there's a Wachtel spring, I call it, is here, and there's a fawn spring is out here. There's also springs that feed the floodplain ponds. We did some gravel culvert replacements in 2019, and that kind of changed the hydrology a little bit, but certain places dried up and certain places fill in with water and retain water longer now. And then we're at the top of I wouldn't say the top, we're at the upper part of the Klamath River estuary. And so all this river up in here is still intertidal. When the tides fluctuate, the river goes down and comes back up on a daily basis. Next slide. So the third stage was to develop the Wetlands Program Plan, the WPP. It's a lot less technical and less involved than the QAPP. And what you do is you come up with some actions that you want to include in your plan that have to correlate to the EPA's core elements framework, or the CEF. The plans by EPA is limited to 6-year durations, so you're not doing a 30-year plan or a 50-year plan. You're doing a 6-year maximum plan, inclusive of the year you start and the year you end. You send a draft to EPA, you get revisions. Again, it takes a long time to get it back. EPA approves it, and then they sign it, and then you have the tribal council approvals, and they sign it, and then it becomes an active plan. Next slide. So some of our actions that we put in the plan were to maintain, actually I think we have 12 in ours. You can have as many as you want. There's no limit on that number of actions, but we try to keep it simple so we can actually achieve the plan and do the actions. One of the actions was to maintain our CRAM assessments, which correlates to core element 1 in that core element framework. So the EPA, there's 1 through 4, and so I just put in the plan which core element we're inside of for each action. So the CRAM stands for, we'll look into that later, but it's a California Rapid Assessment Method used for wetland assessments. One of the actions was to review and refine the QAPP, or the Quality Assurance Plan, because you'll have personnel changes, and there are certain people in the plan that are managers that are responsible for you following the plan and that type of thing, so you might have some changes over time. To maintain our water quality data collection efforts, that's a big one. Continued refinements to bioassessments, that's something that's an ongoing action of the plan. I'll talk a lot about the different bioassessments that were conducted over the last few years and later on in the talk. We have an invasive plants program initiative that's in the plan, and engagement, outreach, and community education is in the plan. Next slide. So we kept it to 12 doable actions. Capacity development, conduct botanical surveys, conduct algae sampling, which we're going to be starting more this year, and it's halfway through the plan. Initiate a watershed restoration program for Junior Creek Wetlands Complex, initiate a bullfrog control program, and capacity building through travel and training. If you don't write the travel and training into your plan, then you're not going to be able to use the funding to go to like conferences like this, or get training for your people to do water quality monitoring or whatever you want to send them to. Next slide. So here's the 4 core elements of the EPA core element framework, and each action should be tied to 1 or more of these parts. A lot of ours are tied to 1, our core element 1, which is monitoring and assessment. That's the easy part. Next slide. I took this out of the core elements document. I think it's 48 pages long. You can download it online, and it basically just kind of defines some processes and how you define wetlands. Next. So on the CRAM assessments, that's I think actually one of our plan. We want to continue doing this. In 2018, we did myself went and did CRAM training. When you do the training, you do about 9 or 10 sites in a week with a class of about 3 practitioners teaching you, and there were about 15 student surveyors. And in 2020, we wrote the cram surveys into the plan. In 2022, we took a second cram training session when we got a new field technician, and we did another 9 sites, I think, in a week with this different practitioners, but the same group that was teaching the cram training. And then 2023, we did some cram surveys at some of our wetlands with a fisheries planning contractor, and the scores are being used in the tribal fisheries habitat restoration plan, which is going to be completed in 2024. And in 2023, we also attended an additional cram refresher course where we did 2 more. I guess I messed up on that. We did 2 more cram surveys with the practitioners. And so cram is an ongoing action in our WPP, our Wetlands Program Plan. Next slide. So water quality monitoring is another part of our plan. We do permanent, we have permanent in-water onset probes that do water level in the ponds, temperature every 15 minutes. It requires downloading monthly to a shuttle and then taking the data into the office and drafting it out. We have dissolved oxygen probes that are continuously monitoring dissolved oxygen in some of our ponds. They're downloaded monthly with the shuttle. We established transect lines in our larger ponds that at each transect every month we do dissolved oxygen, temperature, pH, salinity, conductivity at specified points along each one of those transects. So I think there's three transects in our large pond, 2 and 2 other smaller ponds. We use a variety of instruments, a YSI dissolved oxygen probe that does DO, salinity, and temperature, which we've kind of not used for a while because we've got a new YSI Pro Series water quality meter that does dissolved oxygen, temperature, salinity, pH, etc. And we use another Manta probe from Eureka Incorporated that has a Bluetooth connection to a tablet, and that does turbidity, temperature, and we use that mostly for our CWA surface monitoring sampling. Next slide. So wetland bioassessment projects, we've done invasive species mapping. We started that in 2018 and it's ongoing. We do a spring migration bird point counts, which EPA has a protocol, and we follow that protocol. I do a daily bird list. It's not daily, but it's when I'm sitting in my office, I can see out the window and write down a data form. And then amphibian diversity studies and breeding monitoring is since 2018 ongoing. We do fish diversity studies. We collect macroinvertebrates. We have, I don't know, I think I have about 300 vials now of things to identify. We did a dragonfly diversity study that took us, well, basically, I'll talk about that in a minute. We did wildlife camera studies and botanical surveys and put together a herbarium so that future people can come and see what kind of plants we had before, and then when climate change happens, they can compare and see if those plants are still there or not. Next slide. So here's the invasive species detection and mapping section. We do plants. We have a lot of invasive plants: reed canary grass, Himalayan blackberry, jubata grass, which I think is also called pampas grass, ivy, etc. We have American bullfrog invasive populations. We have invasive mollusks, both freshwater and terrestrial, including the European garden snail, milky slug, a whole bunch of European invasives. We basically just identify those and map out where we see them. And then fish, we have lots of invasive species of fish. We also have native fish. We'll talk about that. But inland silversides, green sunfish, pumpkin seed, black bullhead, yellow bullhead, fathead minnow was a new one since last winter's flooding. Largemouth bass has been in there for about 10 years, I think, 12 years. And we just discovered a smallmouth bass that came in from last winter's flooding as well. So that's a lot of invasive species of fish. In the future, we're now developing a fisheries restoration plan, fisheries habitat restoration plan, and that will probably involve removal of some of these fish if possible. And we also have a lot of native fish, but we're going to talk about that later. And then birds, we have, of course, European starling, English house sparrow, and Eurasian collared dove. There's something really we could do about that. And they're pretty naturalized all across the U.S., all those species. Next slide. So here's a picture of some non-native snails. In 2018, when I started to collect this stuff, there's actually 3 species here, but I started to send it off to experts on snails, and he would identify stuff. Thank you. And so all 3 of these species, the 2 on the upper corner, the middle one, and the 3 on the bottom are invasive. Well, they're non-native. They're not necessarily invasive. They don't do a lot of damage, I don't think, but they are in the system. And we also have some native snails, but they're mostly in the river. We also have native freshwater mussels, 2 species in the river. But we also have a non-native Asian clam, it's called, in the Klamath estuary. It's pretty extensive in large numbers. And next slide. So we also do the spring migration bird point counts according to US EPA protocol. It's important to have a protocol and follow it, or you're not going to be able to compare your data to other studies and year to year. So we use their protocol, and this is some of the stuff in their protocol. We do 5 mornings between May and June 21st in the spring. That's not part of the protocol, but that's just the capacity that we're able to do. And each point count is 5 minutes. You use stopwatch. You record all the birds that you and your partners see at that point for 5 minutes, and all the calls, all the songs, and all the visible birds, even wing sounds, if you can identify that. The points go from, or the surveys go from sunrise to 10 a.m. After 10 a.m., that's part of the protocol. The birds, it gets too warm, and the birds kind of settle down and don't call as much. They're still calling, but they're not as much. And so we stop it at 10 o'clock. If it's really great conditions, we might go till 10:30, but usually we stop at 10. It takes expert birders with years of experience and familiar with the calls and songs and identifications. You just don't want somebody who likes to look at birds to go out there with a field guide and say, oh, I saw this, I saw that. We count all the birds. I already talked about that. We have a standardized data form with the weather, the point number, the location, and what birds we see in that 5 minutes. And if we do record birds that are pre-point and post-point, but we don't count those in. We wouldn't count those in an analysis for the point data. And for consistency, we use the same points every year, and if possible, the same with observers. But last year, we weren't able to have our great bird guy, so we had 2, me and another secondary bird guy. Next. So here's we're collecting this baseline data for pre-dam removal, post-dam removal studies, and maybe we can compare the data when the dams come out if we can continue our surveys. Here's a map of where all our points are. Each point is considered an independent sample, being 100 meters or so apart. So you're not counting the same bird at the same 2 different points. Next slide. So we also have done some wildlife camera studies, and this is one of our latest. We did 15 weeks for wetland associated cameras, and our targets were beaver and otter and mink, because we've gotten those species in our previous beta camera stations. But we shifted over to more wetland sites instead of all. I'll show another map with all the sites of the previous 5 seasons that we did. So the orange triangles are where we got beavers working on our cameras, and we had at least 2 adult beavers and 2 baby beavers in the beginning of the season. And over here in Waukel, number 3, I think we have a different family of beavers. And then the river otter is the purple diamonds, and then we found 2 latrines of otters where they continually come back and use the same spots, and those are in red. Next slide. So here's a picture of our previous, our baited camera stations. We did 5 continual, not simultaneous, 5 seasons in a row. And our seasons are 5 weeks each, which is about triple what the US Forest Service protocol is for Humboldt martin. We were originally doing these baited camera stations trying to determine if Humboldt martin, which is a listed species, occurs on the Rancheria, because our Rancheria is between Patrick's Creek State Park and Blue Creek, which is on the Klamath and the Yurok Reservation. And we're kind of in between, so we were wondering if the Humboldt martin was going to utilize any of our forest types. This area here, 1 through 4, is the redwood hardwood mix. It's late mature, not old growth, probably second growth, but it is late mature, and so that's where we expected to find the martin if we did have it. The red dot is just a security camera that we put up that did get some animals on it. And the red lines are drivable sand and roads, and the purple lines are the trails that we would have to hike to get to the camera stations. So it doesn't show any, this is just where the camera stations were for the first 5 seasons. And by season, I mean inside the season. So March 21st is the first day of spring. We put our cameras in April and May, and not on the edge of the season, but inside the season for 5 weeks, if that makes any sense. If June is 22nd is the first day of summer, we don't put it in on the 19th of June, we put it in, July and August. So we're in the summer, not at the edges of the seasons. Next slide. So in the fall 2021, our focus was on detecting the listed Humboldt martin, the Martes carina humboldtensis. We did not find any of those. We conducted 5 consecutive seasons using basically the same protocol, 5 weeks per session. The U.S. Forest Service uses 18 days per session for martin, so we were trying to overdo it in case we would get them later in each season. We used baited closed set camera stations, and we used a Gusto, which is it's an attractant scent lure that's made out of, I think, skunk guts, I'm not sure, and chicken drumsticks nailed to the bait tree, and the camera is on one tree pointing real close to the bait, and so when they come in and they smell the bait, then they trigger the camera. So we didn't get any martin, so our study became more of a forest carnivore community, and I misspelled prey, a prey diversity study, and 11 species of forest carnivores were detected, and many prey species, and we mapped 135 wood rat nests of prey. That's because we were already there, and we had the GPS and seeing that. So there's a lot of prey out there. It's probably great for martin, but there's also 11 other species of carnivores, and there's sometimes that will deter the martins from coming into the area because there's just so many other carnivores looking to eat them, including the Pacific fisher, which we did have on our study. Next slide. So we got 11 species of carnivores: bobcat, Pacific fisher, black bear, which had distinct seasonality and only showed, there's a few in the fall, nothing in the winter for black bear, and then all of a sudden there's a spike in the spring, and then a real spike of double spring numbers in the summer. And we did that for 5 consecutive seasons, so we see that the seasonality reoccurs in the fall again. Raccoons, striped skunk, long-tailed weasel, river otter, mink, gray fox, coyote, and finally, after 2 years of effort, we got one mountain lion. And then the prey species detected, there's a lot of prey out there for the carnivores: wood rat, brush rabbit, siskiyou chipmunk, which also is seasonal. It hibernates in the winter, you don't have any on your cameras, and then in the spring and summer you start seeing them again. Gray squirrel, northern flying squirrel, deer, mouse, deer. And then in parentheses I have Pacific jumping mouse, Pacific marsh shrew, and Pacific shrew, and long-tailed weasel. that were not on the cameras but were found on minnow traps or different methods. And other species detected. This is not a complete list, but great blue heron, wood duck, mallard, red-tailed hawk, red-shouldered hawk. We have pictures of the red-shouldered hawk flying up, grabbing the bait. The chicken is nailed to the tree. He's trying to get the bait off the tree. It just isn't strong enough. But when the red-tailed hawk came to the same spot, he's able to pluck it off the tree. So that was interesting. Seller's jays, moths, pygmy owl, and we got American beaver on our camera. So that's kind of one of the target species we switched over when we went to the wetland cameras. We were targeting otter, beaver, and mink. Next slide. We also do the fish diversity monitoring. We use bait and minnow traps. And this is in order to inform the fisheries habitat restoration plans. We use spike nets. I don't have a picture. I had a picture of those, but I wasn't able to get it on here yet. Hook and line sampling, that's what we're doing here. And next year, we hope to use some electrofishing methods. Next slide. So we also do amphibian monitoring, and these are the species we have present on the Resighini. I might have left one or two off, I can't remember, but you can see we have quite a diversity of carnivores, mammals, mammal prey. Now we have quite a diversity of amphibians. I don't have a reptile slide, but we have all the reptiles that could be there, except for maybe a western skink and a rattlesnake. So northwestern salamander, northern red-legged frogs, it's a state special concern. So is the foothill yellow-legged. We don't have a lot of those, but we have some breeding and a couple of metamorphs over the years. Northern rough-skinned newts breed there. Coastal tail frogs, also a state special concern, and the southern torrent salamander, which is state special concern. We have the common Pacific tree frog, the noisy one, being the salamander, Del Norte salamander. It's an endemic to the Redwood area. Coast giant salamander, wandering salamander, California slender salamander. Next slide. So here's just a picture. I don't think I put enough pictures in this presentation. But here's a minnow trap with some bullfrogs. One night of bullfrogging. I think we got up to, I want to say 100 and something bullfrogs in the bike net one night, and the next night maybe 75. And our bullfrog removal project, we've taken out about 2,300 in the last 3 years, of mostly tadpoles. So when we do get a metamorph or a juvenile like the one here, we anesthetize those and cut them open and see what they're eating. And we have found that they're eating native fish, non-native fish, and native amphibian larvae. One of them had a bird in it. One of them had a vole, which is like a short-tailed mouse type of a thing. Lots of pollinating insects. I think 15% of all the 105 that I dissected so far had some kind of pollinating insect, which is now becoming kind of an issue. Next slide. So we also conducted a dragonfly diversity study. I started collecting samples in 2018 with just as part of the bioassessment. I was going to try to identify them, the larvae, using some hooks. And then I attended a symposium at the Siskiyou Field Institute where James Johnson was presenting, and I asked him if he was interested in identifying my samples, and he was. So I sent them to him. He found 16 species, and I sent him some more, and he found another one. So we had 17 species of dragonflies in our 450 acres. And then he came and made 2 field visits in 2022, and he brought the total up to 27 verified species of dragonflies and damselflies just on our reservation. And we're planning to publish a checklist, but we don't know. And then you could look at all his pretty pictures, close-ups, and look at his GPS locations at Western Odonata. That's the subclass of dragonflies. And there's a website called Western Odonata. And if you go on there, you can see he's got some marvelous close-up pictures of all the species with the scientific names. I don't have a list of them here, and I can't pronounce all those names anyway. Next slide. But stuff like this, but he's got even closer. That's him holding one. He's got even closer, more beautiful, detailed pictures on that website, Western Odonata. Next slide. So in summary, we collected data towards informing decision-making for restoration projects. We restored some wetlands. I don't know if anyone saw my talk yesterday about cleaning up trash and vehicles and stuff, and that, in that case, we restored a couple of wetlands that were trashed and had trash in them, and we were concerned that toxins would get into the water, surface water and the groundwater. We collected a large mass of data that could potentially be used as pre-dam removal baseline for future studies, and we're working towards invasive species removal, namely bullfrog, and now we have some new staff that are going to focus more on invasive plants and restoration of getting rid of those. We have several species and a lot of acres covered with invasive plants. And the Resighini Rancheria contains a wide variety of habitats, very high biodiversity across many taxonomic species groups. Thank you. Next slide. And so this is just a bunch of quick pictures of different places. This is our biggest floodplain pond. We've fight netted turtle, there's beaver in there, there's muskrat, there's the native turtle, the western pond turtle, largemouth bass, all those invasive species of fish pretty much are in here. Next slide. And here's my coworker, Tyler Jones, who's setting up one of those wetlands transects. Put a rope across the transect. There's rebar established points on each side of the creek, on the pond. So we stretch the rope and then we know where we're going to stay on that transect and do the 5 points across there with all the water quality measurements on each point. So it's like, I don't know, 300 pieces of data a day on that. Next slide. Not every day, but once a month. Here's, I think that's me netting some bullfrog larvae in that same area. Next slide. This is just some pictures of the wetlands. Oh, wait a second before you switch. So just some of the aquatic diversity here for plants. We have the American pond lily. We have 2 species of Potamogeton, which is river weed. We have an invasive called Elodia, which is the goldfish grass that you can get in your aquariums. It's mostly from Brazil, but there's a lot of it out there. We have parrot feather, green algae, filamentous green algae. The list goes on. Cattails, bulrush. Some great healthy wetlands as far as the diversity goes. Next slide. So on pond, this is some of our floodplain pond studies that we do. In each pond, we're doing water quality monitoring. We talked about that. We're doing the invasive species. We're doing the camera studies, and we're doing the birds and stuff when we're out there. I'm keeping the bird list. So that's just at each pond, it's the same kind of treatment. Next. Pond 3 is a smaller pond. We do the same kind of things there. We have cameras there as well. Next slide. This is another shot of some of the invasive aquatic vegetation. Oh, there's one. I think that I didn't notice it, but there's a minnow trap right here in the middle. You can barely see it in the water. It's kind of how we set our minnow traps. It's better to set them so that there's a little oxygen, like some of it's out of the water. So if you get like a frog or salamander or a turtle, they can breathe and not get drowned. But usually you don't leave them in that long, like less than 24 hours, maybe 15 hours, and then you check it. Next slide. This is just the final part. I'm just looking at some specific sites here. This is our culvert that got replaced at the gravel pit. There was a culvert that was blocked here before, and it was like more of an isolated pool, and now it's connected to the whole Junior Creek complex, and probably a mile up from the mouth, the coho will actually get all the way up into this in the last couple of years since we replaced that culvert. Next. Another view of another place with a lot of green algae that they were talking about in a previous talk in this room a couple hours ago. They had this algal blooms. Next. And we did we had a contractor come in and do a bathymetric survey on each of our ponds as part of our fish habitat restoration planning. Next slide. Similar. Next slide. So I'd like to thank the EPA Office Region 9 Office with Loretta Vanegas as our project officer. I'd like to thank the Tribal Council and staff at the Resighini Rancheria, and I list those out: Alicia Dowd, Frank Dowd, Vaughn Murphy, Kendra Jones, Kathy Dowd, the former Natural Resources Directors, Madison Flynn and Shauna McCovey, who is now our present Intergovernmental Affairs Officer. Our new director, Hans Boyd, here with us, and Tyler Jones, who I work with every day. And Dr. Brian Schumacher was really, he was at the EPA, and he really pushed to get my QAPP finalized and approved. It's kind of a complicated and long process, and so I put him in here. And Thomas Kirk of Green Coast GIS, he did a lot of work with me on the wildlife camera studies for the last 2 years, and bullfrog removals as well. And BIA funded several of our first part of our camera studies. And I thank Hans, Lori Harvey, and Michelle Kunst for extensive helps with rewrites and editing on this. I like the color scheme that they put on here too. Next slide. This is the Resighini Rancheria, Bureau of Indian Affairs, EPA Region 9 conference people here, and the EPA overall. Next slide. And any questions, and there's my contact information if you're interested in any specifics on bullfrog removal or any of that stuff.
When you mentioned the turtles, is it the western pond turtle?
Yes. They've done genetic studies on the pond turtles of the Pacific Coast, and there's now a northwestern and a southwestern, and they were considered subspecies. Now they're considered separate species. So we have the northern pond turtle, and they're not common. I mean, we're really shy and hard to see anyway, but we've seen them in our ponds, and we caught them in the fight net a couple times. And we do believe that they're breeding because I found a couple of egg nests that were either predated by raccoons or they already hatched out. I just had the shells in the sand. And so we have a small population, but they're there. And I did see a non-native turtle also, but I haven't seen it for a couple of years. And that was just one specimen, so we're not sure if we still have those or if it was like 50 feet away and it had red on the side of its head, so I can't really say it was. I think it was a red-eared slider, which is non-native and invasive, but we do have the northwestern pond turtle. Anybody else?
Hi, I'm with the Summit Lake Prairie Tribe in Nevada, and I heard that you said that one of the invasive grass species that you're looking at treating is reed canary grass, and we have a very bad problem with reed canary grass, and so I wanted to ask what is your plan, if you do have a current plan with treating that in the future?
We are working on that. Right now we pull it out by hand and try to get rid of it in certain places. Hans, did you want to say something about it? Yeah, we have a really bad problem with it too.
There's several methods to suppress it. It's pretty impossible to get rid of entirely. One of the primary methods we're going to use is revegetation after different restoration projects. If you can establish a canopy, especially a coniferous canopy that's year-round, that will really suppress the growth that grass takes off when it's direct sun. Another strategy is increasing the water depth. If it's invading, pools, you can deepen. The grass can't grow as vigorously if the water is deeper. One of the interesting aspects of the wetlands on the Junior Creek corridor, so close to the estuary, is that there's no salt water. It's completely fresh water, and that's advantageous for coho over wintering. But salt water is another tool that you can use to kind of control reed canary grass in other settings. in other projects I've worked in the past in Humboldt Bay, for example, utilizing some tidal water into channels is another way you can control it. But we are working with some fisheries restoration engineering firm that's coming up with a Great Canary Grass overall control plan that we'll be trying out. So, like I said, it's more of a controlling, kind of knocking it back versus getting rid of it.
Well, thank you, all 25 of you that came to listen. Appreciate it.